Hello everyone, we are Jaded Blanco, Ana Bueno, Jose Anaya, and Mariana Ortiz. In this video, we are going to talk about non-viral vectors, liposomes.
Liposomes are not viral vectors made of lipid bilayers, similar to cell membranes. They can carry genetic material such as DNA or RNA. These systems are biocompatible and biodegradable, and they don't use cause strong immune reactions, which make them very safe for medical use. Liposomes enter the cell mainly through endocytosis, and then release their content into the cytoplasm. Even though their transfection efficiency is lower than that of a viral vectors, they are safe or easy to produce. Today, liposomes are widely used in gene therapy, vaccines, and cancer treatment.
Advantage of non-viral liposomal vectors. One, a greater safety. Liposomal vectors are safer because they don't come from virus. They don't cause infection and produce a much lower immune response, making them better for treatments that need to give more than once. And two, no gene damage. These vectors deliver the gene without insert it into our DNA. That means there's no risk of damaging healthy genes of causing unwanted mutations. Three, can carry more genetic materials. Liposomes can transport large pieces of DNA or RNA than viral vectors. This makes them useful for modern techniques like CRISPR or mRNA therapies. And fourth, easier and cheaper to produce. They are made using simple chemical methods and not living cells. This makes them faster, cheaper, and safer to produce in large amounts. In 5, we can find can be used many times because liposomes don't cause strong immune reactions. The same treatment can be repeated if necessary, and that's harder to do with viral vectors, which often trade immunity after the first dose. In seeds can be designed for specific targets, and scientists can change the compositions of liposomes to make them go directly to certain tissues or organs. This helps the therapy reach where it's needed and reduce side effects.
Today, we are going to talk about the disadvantage of the liposomes. The first problem is making liposomes cost a lot of money. This is very expensive. Liposomes also break down very easily and do not last long. The fat that makes liposome can break down from the heat and the oxygen. The second problem that we have is the delivery efficiency issues. The second problem, the liposomes do not work as good as a virus. They cannot get into the cells well. When they enter to the cell, they get stuck inside. They cannot reach the cell nucleus where the gene must go to work. And the third problem is the blood. Liposomes make the proteins that damage them. The body sends white blood cells to find and destroy these liposomes because they look like invaders. The liposomes die before they can help. It's very quickly. The fourth block and the last one is the immune and cellular challenges. The fourth problem, the body thinks that the liposomes are bad things and attack them. They can hurt the patient. Also, the liposomes can be a little D potions cells. When we add a code to help liposomes live longer, they cannot enter as well to the cell. These are the main problems with the liposomes. Scientists are working to fix these problems and make them better. Well, continue with other disadvantage. The, we are going to talk about the five problem that is liposomes don't find the right cell. They go to the wrong organs instead of the sick cells. This wastes the medicine and doesn't help patients. The sixth disadvantage is the genes from the liposomes don't work forever. After some months, the effects become weak. Patients and need treatments again and again. The seventh problem is few approved products. Not many liposomes treatments are approved by health authorities. It's take many years and a lot of money to get approved. Very few companies want to invest. The eighth problem is the manufacturing problem that making many liposomes at once. It's very difficult. Small batches works well, but big batches have problems. It is expensive and hard to make them all the same.
The use of liposome as non-viral vector has become a very promising strategy in cancer therapy. Unlike viral vector, liposomes are non-living and biocompatible, which means they can safely deliver therapeutic material without causing immune reaction. The structure made of lipid bilayers, like to cell membrane, allow them to encapsulate DNA, RNA, proteins, or drug, and carry them directly into target cells. In cancer treatment, liposomes play an important role in stimulate the immune system. They can activate both EMHC class 1 and class 2 pathways, helping the body to produce cytotoxics T, lymphocytes, and antibody that attach the tumor cells. Cationic liposomes, which has a positive charge, are especially useful because they can attach to negative charged DNA to form lipoplexes, improving gene transfer into cells. Some clinical applications include alobectin 7, a liposomal therapy tested for melanoma, and beta deliver for glioblastoma, both showing encouraging results. To make them more effective, researchers modified their surface with PEG, a process to call it PEGylation, which help liposome circulate longer in the bloodstream. Synthetic system used to transfer genetic materials, DNA or RNA into cells, both seek the same overall goal to get therapeutic genes into cells without using modified viruses, but they using different forms and materials. Liposomes are small spherical vesicles made of lipids, very similar to cell membrane. They have a double lipid layers that can encapsulate DNA or RNA inside. Their main advantage is that they fuse easily with cell membrane, allowing the genetic materials to enter the cell naturally without using viral proteins that could cause immune reaction. In gene therapy, cationic liposomes are the most common because their positive charges bind to DNA and RNA, which are negative charged, and forming lipoplexes. Liposome-based vaccines are a modern and safe approach for improving how our body responds to like a sick. Unlike viral vectors, liposome don't trigger unwanted immune reaction, which makes them much safer. Once inside the body, they can activate both antibody production and cytotoxic T cells, creating a strong and balanced immune response. Because of this advantage, liposome-based vaccines are being widely studied for use against infection and different types of cancer.